So now we're getting to the mechanical part, okay? So here we have some red stuff. Here they tell you they're not going to ask you any specific stuff about specific enzymes. But I and they are going to use specific enzymes as examples of the principles that you've learned. And so I'm going to do the same thing here. Here's an enzyme. You see its ASE ending, sucrase. And it's a good name for an enzyme that helps sucrose split apart into these two things. What kind of reaction did we learn was a splitting reaction? Hydrolysis. So water is also involved here. These are the reactants. Since a bigger disaccharide molecule, see how we review this as we go, is broken into two smaller monosaccharides, this would also be a catabolic reaction, right? Which is going to release a little energy. We're not going to really focus on that right now, right? We're focusing on the mechanics. What does this enzyme do to these two reactant molecules that makes them react? Well, first we got a little vocab that I don't know why somebody thought this was necessary, but some chemist at some point said, you know what, when we're talking about enzymes, we're going to call the reactants a different name maybe, and that's substrate. Substrate is a synonym for reactant, same thing, right? So what does this enzyme do? It actually binds to the substrate. When it binds to the substrate, it's called, for a split second as we'll see, an enzyme substrate complex, which they promise they won't ask you like define that, but it's hard to talk about this without using the term. And you should know that term as an honors biology student. So in a little graphic coming up soon, you'll see that spelled out as well. So here we go with a little more detail. So this is a graphic, right? Which one of these two is the enzyme? Well, hopefully you can use some logic in your knowledge of molecules and say, well, if one of these is sucrose, a disaccharide, and one is a enzyme protein polymer made up of a whole bunch of amino acids, this bigger one's probably the enzyme, and you'd be correct. This is the enzyme. This is the substrate, which is sucrose in this specific case, right? And you can see what obviously happens here. These fit together like two puzzle pieces, a little one and a big one in this case. The part of the enzyme where this attaches, connects, where the action happens, so to speak, is therefore appropriately called the active site. And like two puzzle pieces, it is very specific for sucrose only. Glucose couldn't fit in here. Other molecules couldn't fit in here, and this is where the specificity of enzymes comes from, their shape. One other aspect that I'll bring up in just a minute, but their shape and the shape of the substrate are the biggest key to them being able to help catalyze a specific reaction and not all reactions. Heat, by contrast, was a general catalyst. When you set yourself on fire, you made all this chemistry happen faster, not just specific. Now you notice another important part of how this works. When the substrate fit into the active site to form this enzyme substrate complex, this would be called, a complex is two or more things stuck together, there was a little shape change. You can see this enzyme is kind of squeezed down on the substrate, and what you can't see is that the substrate is actually, in this case, it's being bent a little bit. This aspect, somebody decided long ago, this aspect of how enzymes work that involve a fit where there's a little shape change is referred to by the term induced fit. It's not in your EOC syllabus, but you're going to bump into it, and there you just did. So, shape is a key, but in this active site, I'm going to make a very simple diagram of an enzyme here, and here's the substrate, right? That obviously is going to fit into it because of shape, but there's also always some charges due to a little unequal sharing of electrons, right? Polar covalent bonds, and often in both the substrate and the enzyme. So the way I've drawn them, you can see that what's also going to help this substrate fit in here is a little electrostatic attraction of positives and negatives. So both the shape and the charge are keys to the specificity of enzymes. Okay, so now I threw this in here. This is really an astonishing number. Things at very big scales, like how many stars are in the universe, and things at very small scales like this are hard for we humans to fathom. But here's what turnover number means. A single enzyme molecule like this one can help make an astonishing number of reactions happen faster. That is called its turnover number, and the number is, like I said, astonishing. Thousands and sometimes millions of reactions per second one molecule can make happen. Hard for us to fathom. Now back to how they help these reactions happen, and this will answer this question: can an enzyme be used more than once? So here's a good little diagram. Here's clearly an enzyme. Purple is almost always the protein color in these diagrams. Here's an enzyme, obviously. Here's its active site, right? Here is sucrose, the substrate. We talked about chemistry being part of collisions, right? And the first collision involved in this chemistry is the substrate has to bump into the active site. But if you have a whole bunch of these enzymes, like you would in a cell, and a whole bunch of these, and I mean millions and millions of sugar molecules, sucrose, then you're going to get a bunch of collisions of substrate into the active site to make that thing that I told you, enzyme substrate complex. And what they don't show too well here, which they did in the other one, is this enzyme changes shape a little bit, and this substrate gets bent. Where does it get bent? Right at the place that's going to break. It's almost like if you were the enzyme and you were holding a stick like this, holding a stick, and you changed the shape of yourself so that it bent right there, you'd make it more likely to break right there. This is mechanically what we understand enzymes do to their substrates that they're going to break in catabolic reactions. They physically bend them. Now, what's going to make this bond break? It's not the enzyme bending it. It's a collision, because this is being bumped into by lots of things. But without the bending of this enzyme, the collisions that would normally happen in a cell to this sucrose are not strong enough to break this bond. But if it's weakened because it's bent, then these same collisions can make it happen. What's another way of thinking about that? The strength and number of collisions is also due to temperature, how fast the particles are moving, which is the definition of temperature. And so that means at normal temperature and normal collision number and speed, not much of these are going to break apart. But with the help of an enzyme, at that same force of collision, it's going to happen faster. That makes sense? It should. Now, this little diagram doesn't emphasize the role of water, but as we said, this is a hydrolysis reaction, right? This water molecule also gets split. This water molecule also has to fit into the active site, which they really could have showed here. But here's where the water molecule ends up. Part of it attached to glucose, part of it attached to fructose. These are the products, right, that this reactant got changed into with the help of this enzyme. Collisions made the reaction happen. The enzyme helped make it happen faster, and it's very specific. This enzyme could not make maltose, for instance, is another disaccharide. This enzyme couldn't help break up maltose because maltose doesn't fit. It's a little different shape. So now, how does this enzyme make anabolic reactions happen faster? Well, in class, we might do a little exercise here, but without doing that little exercise, here we go with this. I'm going to draw another diagram of an enzyme, and now I made the active site like two shapes worth. So here we're going to have two substrates. My art's not great. That fit into here, and this reaction is going to involve these two reactants connecting to each other, an anabolic reaction. right? Well, how this makes anabolic reactions help is that these two molecules, if you think of these as two amino acids, for instance, if you remember way back, when amino acids combine, it's the carboxyl of one that bumps into the amine of another, and that peptide bond forms. So if they bump into each other in any other way than carboxyl and amine bumping in, they're not going to react. What does the enzyme do to help? It holds them right in that right position so that what makes the reaction actually happen? A collision, something bumping into these things, forces them now so close together that they start sharing electrons, they bond. So here it is in words. I'm just going to flash this up quickly. You can freeze it and stop it. I've said all these words. How do enzymes work mechanically? They always combine with their substrate in the enzyme's active site. But then what kind of reaction is causes a little different mechanical event in terms of catabolic versus anabolic reactions. If it's catabolic, the shape change bends bonds, puts a stress on those bonds that are going to break and makes them break easier. And if it's an anabolic one, the two or more substrates that are being held in the right position. That makes them more likely to bond. That means the reaction's happening faster. So now, real fast, you learned that the nucleus is the control center of the cell, right? We're going to go into a lot more detail about what that means, because your brain you would identify as your control center probably, and sometimes the nucleus is like made an analogy of the brains of a cell, but it doesn't make decisions quite like your brain. Here's why we have come to call it the control center. What needs to be controlled in a cell? Chemical reactions. What we just got finished saying was life is a bunch of well-ordered chemical reactions, and the specificity of enzymes has everything to do with how they get controlled in a cell. What is found in the nucleus? This nucleic acid molecule called DNA, right? What does it do? Well, DNA is like a recipe book. It's like a chemical recipe book. What's it a recipe for? Proteins, like the enzymes we're talking about. So if I want to make a certain reaction happen, and it's got a specific enzyme, then that story starts in the nucleus, where part of a recipe book that we call a chromosome, that part that we call a gene, is used as the instructions to put the right amino acids together to make this particular enzyme, and then this reaction starts happening faster. That's enough of an introduction to that. We will spend an entire 2 units on this later.